Hello dear students, as you are preparing for your practical exam, so with this video I would like to help you to present yourself well in your coming exam. So for a practical exam, most important part is your viva, that is your presentation, how you present yourself in front of an examiner. Okay, so most probably you will be asked the questions which are related to the experiments that you will be doing in your exam. For example, the major experiments that you will do is from pollen germination or the preparation of slide, then population density, and then the process of isolating DNA from a sample or any plant material. So you can be asked related to all these topic. Okay, for example, from pollen germination, the entire chapter of sexual reproduction in flowering plant is covered. I mean, from entire chapter you can be asked from any area. For this you have to be very clear understanding about microsporogenesis and megasporogenesis, followed by fertilization and post fertilization events. Okay, here you need to understand the basic technical differences between microsporogenesis and megasporogenesis, or what happens when pollen germinates or how does it germinate. Okay, how exactly the male gamete fertilizes only the egg and not the other adjoining cells, since inside the female gametophyte there are other cells as well, including your antipodal cells, synergids, and also the polynuclei. But particularly how the gamete is capable of only fertilizing the female gamete alone. Okay, and also you should know about the various stages of microsporogenesis, and also both meiosis and mitosis occurring at different stages. So what is their significance, and why they are different, and at what steps they are done. So all these things should be very clear, because both in microsporogenesis and megasporogenesis, both mitosis and meiosis occurs. Since meiosis leads to the production of haploid cells, and mitosis leads to the production of the cell with the same chromosome number. So you should have the understanding about it, the ploidy of the different cell at different stages of microsporogenesis and megasporogenesis. Then also from the part of pollination, the various agents of pollination. Okay, and types of flower, clistogamous, chasmogamous flower, and then about different agents pollinating the flower and their adaptation for pollination. Okay, so these are important. And then from the part of DNA isolation, you should be learning about some of the important processes like PCR, or you can say gel electrophoresis, or the role of various enzymes during the isolation of DNA, and also about the chemical nature of DNA, or the nucleotides. Okay, nucleotides, and since the DNA or the RNA are the polymers of nucleotide bases, so you need to understand their chemical nature, the type of bonding between the different molecules. Okay, so it is important. And as I said, the procedure is also important. You can be asked the particular procedure, why you are doing this. Say for example, why you are using different enzymes, or what are the various enzymes that you will be using during the process of isolation of DNA. Okay, and its significance in the process. So all these things you need to understand and you need to learn. Then thirdly, from the experiment, to find the species richness or the population density of a given area. Okay, for this you need to know the formula to calculate the population size or the population density, and also the scientists who have proposed it, the various factors which is responsible for the species richness of a particular population of a species in a given area. Okay, along with this you should also know about the population interaction among the various groups of organism, with their examples. Okay, that is important. yeah, this is one part of your viva, that is practical plus viva. Then secondly is your project. For project, it is very important that you should know well about your project. Many a times it happens that the students simply copy it from someone or from their friend and simply they submit it. So it should not happen. You should be very clear about your project, since every project has its objective. So you should have your personal understanding, what you have learned from your project, what is its objective and what is its conclusion, and what personally from your own view what you would like to convey to the society, or convey it to the examiner. Okay, so that is very important. So you should be very clear about it. You should not simply be submitting it, but you should go thoroughly of the project that you are about to submit, or that you are submitted already. Okay, that is also one part. And from sporting also, there are some of the area which you need to take care of. For example, the gametogenesis. Gametogenesis including the oogenesis as well as the spermatogenesis. These two here, as you will study on the permanent slide, the process of spermatogenesis and oogenesis, or blastula, the different stages of the blastomeres, like blastocyst you can say. So up to the different stages of blastomeres till implantation, all these stages you need to understand well. And also the oogenesis process and spermatogenesis, and the role of hormones at various stages. Okay, and then the next is from principle of inheritance and variation. Okay, Mendelian genetics, about pedigree analysis, about the different types of genetic disorder that follows Mendelian principles of inheritance, including your color blindness or hemophilia, etc. So basically here you need to know what are the different types and how are they categorized as recessive or dominant or sex linked or autosomal. So you have to know the characteristic identifying features of those diseases. Okay, and also should know how to make a pedigree of a certain disease. Okay, so these are some area which you can study for your practical exam. Of course, as I said, the procedure why you are doing that particular step is very important. Okay, for example, for pollen germination, the nutrient medium, what are the composition of the nutrient medium that you have prepared. Okay, and the different precautions that you will take during the experiment. Yeah, all these things are important. Yes, of course. And as the course or the syllabus is vast, so you can be asked from any area. But as I said, the key area I mean particularly the experiments that you are doing, or the projects that you are submitting, or the spotting, for the spotting the different specimen, like the adaptations in animals and adaptation in plants, which are also there in the theory, are very important. For example, adaptation in mammals in polar areas or in colder region, or adaptation of different animals in a dry region or in a xeric region. So these are also very important. How would you identify, or what are different adaptive features in them? Okay, so these three key areas, as I said: number one, your practical related topic, which is also there in the theory. Secondly, your project, which is very important. Then thirdly, from spotting. Okay, in on spotting also, all the topics are there in the theory as well. So these are the three key areas which you need to be careful, and also which you need to prepare for your practical exam. And here, as I said, your oral presentation is really very important for your practical exam. So that's why I wish and I hope that you will do well. So present yourself well. Okay, so that's it. I hope this video will help you, and thank you so much.